Trash. It's a part of our everyday lives that we don't really pay much attention to it unless we want to throw it away. But why do we throw away our trash? What if we just let it accumulate? Well, the reason we don't do that is because excess waste in our environment isn't good for our health. A similar thing is true in the human body. See, our bodies create a certain amount of waste as a byproduct of cellular processes. If this waste isn't properly disposed of, it builds up in the cells and causes adverse effects on the human body. This usually occurs due to a lysosomal storage disease, which means that the lysosomes, which are essentially the trash cans of the cells, don't function properly, leading to a buildup of wastes in the human body. Neurons, in particular, are quite sensitive to this buildup of waste. When all this waste builds up in the neurons, the brain doesn't function as it should and often leads to neurodegenerative diseases. An example of this is CLN3 disease, also known as juvenile Batten disease. Although there are currently no curative treatments for this disease, the employment of gene therapy may lead to fantastic results in lengthening and increasing the quality of life in those with CLN3 disease. Welcome to Taking Out the Trash with a Cure for CLN3 disease. My name is Suha Koilakh, and I will be discussing how gene therapy can be used to create a potential experimental treatment for CLN3 disease. However, in order to explore potential treatments for CLN3 disease, we must first understand how it is caused. CLN3 disease is a monogenic disease, meaning it is caused by a mutation in a singular gene, in this case the CLN3 gene. In a healthy person, the CLN3 gene is present and codes for the CLN3 protein. This protein is necessary for lysosomal function, and its presence allows the body to keep running smoothly. However, in someone with CLN3 disease, there is an approximately 1 kilobase deletion present in the CLN3 gene. Because of this, the CLN3 protein is non-functional and the lysosome cannot function as it should. This leads to a buildup of waste and leads to neuron death, which causes neurodegeneration. The disease affects juveniles typically from the age of 5 to 15. Before the onset of symptoms, the child will progress normally. However, after the first symptom appears, which is blindness, the child will gradually decline both cognitively and physically. This is then followed by reduced mobility and the onset of seizures. Eventually, the patient passes in their 20s to 30s. CLN3 disease is a horrific disease, affecting not only the children who have to endure these struggles, but also their family and friends who have to be prepared for all of this. The children pictured in this slideshow are actual patients with CLN3 disease. You can read their story on forbatten.org, and I highly encourage it. I will be linking the website in the description. There are many ways to diagnose CLN3 disease. Because blindness is the first symptom, it may first be suspected at an eye exam. A prominent feature in those with CLN3 disease is bull's-eye maculopathy, which is pictured. It appears as a target in the macula, which is a part of the retina and is caused by degeneration of the macula. Tests such as electroretinograms can detect such degeneration and aid in the diagnosis of CLN3 disease. Skin biopsies can also be used to detect unusual buildup of lipofuscins, which are commonly known as aging pigments. They are yellow-brown deposits of fat and proteins that are usually broken down, but in those with lysosomal storage disorders may create abnormally large deposits in skin cells. Still, the findings from these two diagnosis methods may signify diseases other than CLN3 disease. The only way to confirm a diagnosis of CLN3 disease is to get a DNA test. Despite all the hardships that come with CLN3 disease, the only treatments available manage the symptoms. There is no treatment that treats the cause of CLN3 disease and slows progression. The problem remains unsolved. This is why we decided to use gene therapy in our experimental treatment for CLN3 disease. Gene therapies are treatments that alter gene expression, and because of this, they allow us to treat the source of mutations, something that is revolutionary in the field of medicine. Our application of gene therapy focuses on the central dogma of biology, which states that DNA is transcribed into mRNA, which is then translated to proteins, which are essential for a functioning body. CLN3 disease is caused by a deletion mutation in the CLN3 gene. Based off of this, if we reintroduce the gene in the body, it should be able to create the CLN3 protein. However, in order to make this process more efficient, we'll be introducing an mRNA copy of the CLN3 gene instead of the DNA copy. The body will then be able to translate the mRNA into the CLN3 protein, which should create functional lysosomes. However, we cannot simply inject the mRNA into the body and hope for the best. We need to use something called a vector to carry the mRNA to the specific cells that need to be treated. For our purposes, we'll be using an AAV2 vector. We chose this vector because the AAV vector is RNA-based and therefore can carry our mRNA cargo. It can also carry more genetic cargo than we need. In addition, AAV2 is able to transduce cells in the central nervous system quite effectively and efficiently, which is what makes it the ideal vector for our purposes. In order to create the vector with the mRNA, we need a plasmid. A plasmid is a circular strand of double-stranded DNA, and in a simplified description of AAV synthesis, the plasmid is transfected into HEC293 cells, which are human embryonic kidney cells. These cells then express the plasmid's viral proteins with a transgene of interest, in this case the CLN3 gene, inside it. After fully expressed, the product can then be purified and used. To effectively deliver the drug to the brain, we need to bypass the blood-brain barrier. The purpose of the blood-brain barrier is to keep out harmful substances from the brain, but this makes it difficult for any drug targeting the brain. We chose an intracisterna magna injection, pictured below, because it is less invasive out of all the options while still being effective. Other techniques we considered were intravenous injections, though they are unable to bypass the blood-brain barrier. We also considered a cerebrospinal fluid injection, but an intracisterna magna injection would be more effective. Lastly, we considered an intraparenchymal injection directly into the brain, but considering the age range of those affected by CLN3 disease, it would be unnecessarily invasive. In order to create a proof of concept model, we partook in lab work for the necessary steps. Firstly, we cultured astrocytes because of how similar they are to neurons. If these astrocytes were successfully transduced by our vector, it can reasonably be said that neurons would be transduced as well. When the AAVs containing mRNA for green fluorescent protein were introduced to the astrocytes, we can clearly see that the astrocytes were glowing green. This means that the astrocytes were able to successfully express the proteins in the AAVs. This means that if we were to replace the mRNA for the green fluorescent protein with the mRNA for the CLN3 protein, the astrocytes and neurons would be able to express that as well. With a functional CLN3 protein and lysosomes, the body might finally be able to dispose of all the trash that has accumulated over time. Although the treatment will not be able to reverse the disease, it could potentially slow down progression, increasing quality of life for those affected by the disease and allowing them to live longer with their loved ones. I would like to specially thank Kripa, Sudhir, and Mary Bunn for working with me for the first iteration of this treatment, which used a lipid nanoparticle as the vector of choice in an IV injection. We also worked together for the first half of this presentation concerning the overview of CLN3 disease. I would also like to thank UNC Wilmington and NCSSM for making this research and lab work possible, along with all others who worked with us for the proof of concept models. And finally, thank you for watching this video. If you have any questions, feel free to comment or email me at sakgenetics@gmail.com.